everyone. Welcome to a new session on molecular biology. In this session, we will be discussing DNA repair mechanisms. DNA is the repository of genetic information in each living cell. Its integrity and stability is essential for life. DNA, however, is not inert. Rather, it is a chemical entity subject to assault from the environment, and any resulting damage, if not repaired, will lead to mutation and possibly some severe disease. Perhaps the best known example of the link between environmental induced DNA damage and disease is that of skin cancer, which can be caused by excessive exposure to UV radiation in the form of sunlight. Beyond environmental agents, DNA is also subject to oxidative damage from byproducts of metabolism such as free radicals. In fact, it has been estimated that an individual cell can suffer up to 1 million DNA changes per day. Our cells are constantly exposed to insults from endogenous and exogenous agents that can introduce damage into our DNA and generate genomic instability. To counteract the harmful effects of DNA damage, cells have developed a specialized DNA repair system which can be subdivided into several distinct mechanisms based on the type of DNA lesion. These processes include base excision repair, mismatch repair, nucleotide excision repair and double-strand break repair, which comprise both homologous recombination and non-homologous end joining. Since many mutations are deleterious, DNA repair systems are vital for the survival of all organisms. Changes to the cellular genome can generate errors in the transcription of DNA and ensuring translation into proteins necessary for signaling and cellular function. Genomic mutations can also be carried over into daughter generations of cells if the mutation is not repaired prior to mitosis. Once cells lose their ability to effectively repair damaged DNA, there are three possible responses. 1. The cell may become senescent, that is, irreversibly dormant. 2. The cell may become apoptotic. 3. The cell may become malignant. Here you can see a figure explaining about DNA repair. In this, you can see a healthy cell when exposed to DNA damaging agents which may be exogenous or endogenous turn to cells which have damaged DNA. But if the cell possesses DNA repair mechanisms, the damaged cell can turn back and become a healthy cell. If unrepaired DNA damage persists, the cell accumulates the DNA damage and the cell can become either a senescent cell, apoptotic cell or a cancerous cell. To compensate for the degree and types of DNA damage that occurs, cells have developed multiple repair processes. DNA repair is regulated in mammalian cells by a sensing mechanism that detects DNA damage and activates a protein called p53. Endogenous and exogenous damage. Types of endogenous damage. The various damages caused to DNA due to endogenous cellular process are A. Oxidation of bases, B. Alkylation of bases, C. Hydrolysis of bases, D. Adduct formation, E. Mismatch of bases. Causes of exogenous damage. UVA and UVB radiations, ionizing radiations, thermal disruption, industrial chemicals such as vinyl chloride, hydrogen peroxide, polycyclic aromatic hydrocarbons, alkylating agents. DNA repair systems. DNA damage occurs spontaneously and as a result of ubiquitous environmental agents. Most organisms possess some capacity to repair their DNA, and DNA is the only macromolecule which is repaired by cells. The repair mechanisms are divided into three categories: damage reversal, damage removal and damage tolerance. Damage reversal. Photoreactivation. Cells are endowed with repair mechanisms to correct radiation damages and to bring the DNA back to its normal state under the influence of intense blue light. This reversal of the mutagenic effect of UV radiation by visible light is called photoreactivation. The photolyase enzyme catalyzes this reaction, which utilizes light energy to break the bonds between purine dimers. Here you can see the action of photolyase along with white light, which breaks the photodimer into separate thymine molecules. But on exposure to UV light, the thymine residues in turn become a photodimer. Ligation of single strand breaks. Single strand breaks or SSBs arise from direct attack by intracellular metabolites and from spontaneous DNA decay. Simple breaks in one strand are rapidly repaired by DNA ligase. Microbial mutant lacking ligase tend to have high levels of recombination since DNA ends are recombinogenic. Bloom syndrome in humans is involved with DNA ligase deficiency. Let us now study about damage removal type DNA repair mechanisms. To compensate for the many types of DNA damage that occurs, cells have developed 5 major DNA repair mechanisms, which are base excision repair or BER, mismatch repair or MMR, nucleotide excision repair NER, and double strand break repair, which includes both homologous recombination HR and non-homologous end joining NH-EJ. In this table, you can see the 5 different types of DNA repair mechanisms and the important genes which are involved in the same. Base excision repair or BER. Base excision repair is primarily responsible for removing small non-helix distorting base lesions from the genome. BER is initiated by DNA glycosylases, which recognize and remove inappropriate bases forming apurinic site or AP site and are cleaved by an AP endonuclease. DNA polymerase then removes the damaged region using its 5' to 3' exonuclease activity and correctly synthesizes the new strand using the complementary strand as a template. The resulting single strand break can then be processed by either short patch or long patch base excision repair method. Short patch method. In short patch method of BER, a DNA glycosylase cleaves the bond between the nucleotide base and ribose, leaving the ribose phosphate chain of the DNA intact, but resulting in an apurinic site. 8-oxoguanine DNA glycosylase 1 or OGG1 removes 7,8-dihydro-8-oxoguanine or 8-oxo-G, one of the base mutations generated by reactive oxygen species. Polymorphism in the human OGG1 gene is associated with the risk of various cancers such as lung and prostate cancer. Uracil DNA glycosylase, another base excision repair enzyme, excises the uracil, that is the product of cytosine deamination, thereby preventing the subsequent cytosine to thymine point mutation. N-methylpurine DNA glycosylase or MPG is able to remove a variety of modified purine bases. The AP sites in the DNA that result from the action of BER enzymes are repaired by AP endonuclease 1 or APE1. APE1 cleaves the phosphodiester chain 5' to the AP site. The DNA strand then contains a 3' hydroxyl group and a 5' A-basic deoxyribose phosphate. DNA polymerase beta or Pol beta inserts the correct nucleotide based on the corresponding WC pairing and removes the deoxyribose phosphate through its associated AP lyase activity. The presence of X-ray repair cross complementing group 1 or XRCC1 is necessary to form heterodimer with DNA ligase 3 Lig3. XRCC1 acts as a scaffold protein to present a non-reactive binding site for Pol beta and brings the Pol beta and Lig3 enzymes together at the site of repair. Poly-ADP ribose polymerase or PARP1 interacts with XRCC1 and Pol beta and is a necessary component of the base excision repair pathway. The final step in the repair is performed by Lig3, which connects the deoxyribose of the replacement nucleotide to the deoxyribosyl phosphate backbone. This pathway has been named short patch BER. Long patch base excision repair. Long patch base excision repair replaces a strand of nucleotides with a minimum length of 2 nucleotides. Long patch BER requires the presence of proliferation cell nuclear antigen or PCNA, which acts as a scaffold protein for the restructuring enzymes. Other DNA polymerases, possibly Pol delta and Pol epsilon, are used to generate an oligonucleotide flap. The existing nucleotide sequence is removed by flap endonuclease 1 or FEN1. The oligonucleotide is then ligated to the DNA by DNA ligase 1 or Lig1, sealing the break and completing the repair. Nucleotide excision repair or NER. Nucleotide excision repair is the most flexible of the DNA repair pathways, considering the diversity of DNA lesions it acts upon. The most significant of these lesions are pyrimidine dimers caused by the UV component of sunlight. Other NER substrates include bulky chemical adducts, DNA intrastrand crosslinks, and some forms of oxidative damage. The common features of lesions recognized by the NER pathway are that they cause both a helical distortion of the DNA duplex and a modification of the DNA chemistry. Mammalian nucleotide excision repair or NER eliminates carcinogen DNA adducts by double endonucleolytic cleavage and subsequent release of 24 to 32 nucleotide long single-stranded fragments. The NER process requires the action of more than 30 proteins in a stepwise manner that includes damage recognition, local opening of the DNA duplex around the lesion, dual insertion of the damaged DNA strand, gap repair synthesis, and strand ligation. There are two distinct forms of nucleotide excision repair: GGNER, which is the global genomic NER, which corrects damage in transcriptionally silent areas of the genome, and TCNER, which stands for transcription coupled NER, which repairs lesions on the actively transcribed strand of the DNA. These two sub-pathways are fundamentally identical except in their mechanism of damage recognition. In GGNER, the XPC or Xeroderma pigmentosum complementation group C protein complex is responsible for the initial detection of damaged DNA. Conversely, damage recognition during TCNER does not require XPC, but occurs when the transcription machinery is stalled at the site of injury. The stalled RNA polymerase complex is displaced in order to allow the NER proteins to access the damaged DNA. This displacement is aided by the action of CSA or Cockayne syndrome A and CSB proteins as well as other TCNER specific factors. The subsequent steps of GG and TCNER proceed in an essentially identical manner. XPA and the heterotrimeric RPA or replication protein A then bind at the site of injury and further aid in damage recognition. Next, the XPB and XPD helicases, components of the multi-subunit transcription factor TF2H, unwind the DNA duplex in the immediate vicinity of the lesion for the enzyme RNA polymerase II to begin transcription. The endonucleases XPG and ERCC1, that is Excision Repair Cross Complementing Group 1 or XPF, then cleave one strand of the DNA at positions 3 and 5 to the damage respectively, generating an approximately 30 base oligonucleotide containing the lesion. Finally, the nick in the repaired strand is sealed by a DNA ligase, thus completing the nucleotide excision repair. 3 syndromes are associated with inborn defects in nucleotide excision repair. They are xeroderma pigmentosum or XP, Cockayne syndrome or CS, and Trichothiodystrophy, which is also known as TTD, all characterized by exquisite sun sensitivity. The disorder arises from mutations in one of the 7 genes, that is XPA to XPG. Cockayne syndrome caused by mutation in the CSA or CSB genes is a TCNER specific disorder that is remarkably dissimilar from xeroderma pigmentosum. No predisposition to cancer is observed because TCNER defect causes CS cells to be particularly sensitive to lesion induced apoptosis, thereby protecting against tumorigenesis. Physical and neurological developments are impaired, resulting in dwarfism and dysmyelination. The syndrome includes features of premature aging, which is related to the increased trigger for apoptosis induced by transcriptional arrest from endogenous lesions in combination with the TCNER defect. TTD is a condition sharing many symptoms with the Cockayne syndrome, but with the additional hallmarks of brittle hair, nails and scaly skin. TTD is not associated with cancer. Mutations in the XPD or XPB genes gives rise to all 3 diseases. Thus, as subunits of TF2H, XPB and XPD have dual functions, NER and transcription initiation. Mutations not only compromise NER, but also affect transcription, causing developmental delay and reduced expression of the matrix proteins that causes brittle hair and scaly skin. TCNER initiates when RNA polymerase stalls at a lesion in DNA. The blocked RNA polymerase serves as a damage recognition signal which replaces the need for the distortion recognition properties of the XPC RAD23B and DDB complexes. CS proteins such as CSA and CSB bind some types of DNA damage instead of XPC RAD23B. Mutations in TCNER machinery are responsible for multiple genetic disorders, including TCNER associated diseases such as Trichothiodystrophy or TTD is an autosomal recessive inherent disorder characterized by brittle hair and intellectual impairment. Cockayne syndrome, also known as Neal-Dingwall syndrome, is a rare autosomal recessive neurodegenerative disorder characterized by growth failure, impaired development of the nervous system, abnormal sensitivity to sunlight or photosensitivity, eye disorders and premature aging. Double-strand break repair. Double-strand DNA breaks or DSBs are most biologically hazardous types of DNA damage and are common events in eukaryotic cells. There are 2 major pathways for repairing them: homologous recombination and non-homologous DNA end joining or NHEJ. These 2 repair systems differ in their requirement for a homologous template DNA and in the fidelity of DSB repair. HR directed repair is largely an error-free mechanism as it utilizes the genetic information contained in the undamaged sister chromatid as a template. In contrast, non-homologous end joining is normally error prone and involves elimination of DSBs by direct ligation of the broken ends. The HR pathway is activated when the cell is in late S or G2 phase and the template has recently been duplicated. This mechanism requires the presence of an identical or nearly identical sequence linked to the damaged DNA region via the centromere for use as a repair template. Double-stranded breaks repaired by this mechanism are usually caused by the replication machinery attempting to synthesize across a single strand break or unrepaired lesion, resulting in the collapse of the replication fork. Non-homologous end joining or NHEJ is used at other points of the cell cycle when sister chromatids are not available for use as HR templates. When these breaks occur, the cell would not have replicated the region of DNA that contains the break. So, unlike the HR pathway, there is no corresponding template strand available. In non-homologous end joining, the Ku heterodimeric protein positions the two ends of the broken DNA strands for repair without an available template, losing sequence information in the process. Multiple enzymes are involved in the rejoining process, including DNA ligase 4, XRCC4 and DNA dependent protein kinase or DNA PK. Non-homologous end joining is inherently mutagenic as it relies on chance pairings called microhomologies between the single-stranded tails of the two DNA fragments to be joined. In higher eukaryotes, DNA PK is required for non-homologous end joining repair both via the primary mechanism and via an alternative backup mechanism NHEJ. NHEJ is critical not only for the repair of pathological DSBs as in chromosomal translocations, but also for the repair of physiological DSBs creating during VDJ recombination and class switch recombination. Therefore, patients lacking normal non-homologous end joining are not only sensitive to ionizing radiations, but also severely immunodeficient. When double-strand breaks arise in any organism, prokaryotic or eukaryotic, there are two major categories of DNA repair that can restore the duplex structure. In this image, you can see the physiological and pathological causes of double-strand breaks in mammalian somatic cells. Mismatch repair or MMR. The mismatch repair MMR system plays an essential role in post-replication repair of misincorporated bases that have escaped the proofreading activity of replication polymerases. MMR proteins also correct insertion or deletion loops or IDLs that result from polymerase slippage during replication of repetitive DNA sequences. The significance of this pathway is corroborated by the fact that MMR deficient cells are said to have displayed a mutator phenotype which is characterized by invariably microsatellite instability and an elevated mutation frequency. The MMR pathway can be divided into three principal steps. A recognition step where mispaired bases are recognized, an excision step where the error containing strand is degraded resulting in a gap, and a repair synthesis step where the gap is filled by the DNA resynthesis. The mismatch repair process is highly conserved from E. coli to humans. The canonical human mismatch repair pathway is carried out by two major protein complexes which are so called MUT-S and MUT-L based on their homology to the E. coli mismatch repair proteins. While MUT-S is responsible for mismatch recognition, MUT-L couples the recognition of the mispaired bases by the MUT-S complexes to downstream MMR events which lead to the removal of the strand containing the error. In mammalians, the initial mismatch recognition step is fulfilled by two MUT-S activities that function as heterodimers. The MSH2-MSH6 heterodimer, also known as MUT-S alpha, preferentially recognizes base-base mismatches and small IDLs of 1 or 2 nucleotides, while MUT-S beta, the heterodimer of MSH2 and MSH3, recognizes larger IDLs. Formation of the MUT-S DNA complex is followed by ATP dependent recruitment of MUT-L homolog or MLH complexes. 3 MUT-L activities have been identified and like MUT-S also function as heterodimeric complexes. MUT-L alpha, an heterodimer of MUT-L H1 and PMS2, which contains the primary MUT-L activity approximately 90% in humans and supports the repair initiated by both MUT-S alpha and MUT-S beta. The 2 additional MUT-L heterodimers consist of MLH1 or PMS2, that is MUT-L beta, and MLH1 or MLH3, that is MUT-L gamma, which may play minor roles in MMR. Assembly of the ATP dependent MUT-S MUT-L DNA heteroduplex ternary complex is necessary to activate exonuclease mediated degradation of the error containing strand. In humans, this degradation is performed by exonuclease 1 EXO1 through its 5' to 3' exonucleolytic activity. The entry point for EXO1, which may be thousands of nucleotides from the mismatch, is generated via single strand excision by the pCNA or replication factor C RFC dependent endonuclease activity. The extensive gap left by EXO1 is then resynthesized by DNA polymerase delta, which is accompanied by at least 2 other proteins, pCNA and replication protein A RPA. Lastly, mismatch repair is completed by DNA ligase 1 sealing of the remaining nick. Let us now study about the last DNA repair system, DNA damage tolerance. All DNA damage cannot be removed immediately. Some of it may persist for a while. If a DNA replication fork encounters DNA damage such as a pyrimidine dimer, it will normally act as a block to further replication. However, in eukaryotes, DNA replication initiates at multiple sites and it may be able to resume downstream of a dimer, leaving a gap of single-stranded unreplicated DNA. The gap is potentially just as dangerous, if not more so, than the dimer if the cell divides. So, there is a way to repair the gap by recombination with either the other homolog or the sister chromatid. This yields 2 intact daughter molecules, one of which still contains the dimer. Recombinational repair or daughter strand gap repair. This is a repair mechanism which promotes recombination to fix the daughter strand gap and is a way to cope with the problems of a non-coding lesion persisting in the DNA. This type of recombinational repair is generally accurate and requires a homolog or sister chromatid. The products of the human breast cancer susceptibility genes BRCA1 and BRCA2 may be involved in recombinational repair together with homologs of the yeast RAD51 and RAD52 genes. A second type of recombinational repair, which is used primarily to repair broken DNA ends, are caused by ionizing radiation and chemical mutagens with similar action as that of non-homologous end joining reaction. This repair system is also employed by B and T cells of the immune system for genetic rearrangements needed for their function. The Cu70 and Cu80 and DNA dependent protein kinase proteins are needed for non-homologous end joining. Mutagenic repair or trans-lesion synthesis. An alternative scenario for a DNA polymerase blocked at a dimer is to change its specificity so that it can insert any nucleotide opposite to the dimer and continue replication. Regulation of damage control. DNA repair is regulated in mammalian cells by a sensing mechanism that detects DNA damage and activates a protein called P53. P53 is a transcriptional regulatory factor that controls the expression of some gene products that affect cell cycling, DNA replication and DNA repair. Some of the functions of P53 which are just being determined are stimulation of the expression of genes encoding P21 and GAD45. Loss of P53 function can be deleterious. About 50% of all human cancers have a mutated P53 gene. The P21 protein binds and inactivates a cell division kinase or CDK, which results in cell cycle arrest. P21 also binds and inactivates pCNA, resulting in the inactivation of replication forks. The pCNA or GAD45 complex participates in excision repair of damaged DNA. Let us now summarize what we have learnt in this session. The DNA in a human cell undergoes several thousand to a million damaging events per day, generated by both external and internal metabolic process. The DNA repair mechanisms is divided into three categories such as damage reversal, damage removal and damage tolerance. To compensate for the many types of DNA damage that occurs, cells have developed multiple repair mechanisms wherein each corrects a different subset of lesions. Mammalian cells utilize 5 major DNA repair mechanisms: base excision repair BER, mismatch repair MMR, nucleotide excision repair NER, and double strand break repair, which includes both homologous recombination HR and non-homologous end joining NHEJ. The biological significance of DNA repair mechanisms is underscored by the fact that their deregulation can contribute to the initiation and progression of cancer. On the other hand, DNA repair can confer resistance to frontline cancer treatments, that is chemotherapy and radiation, which rely on the generation of DNA damage to kill cancer cells. Thus, the sensitivity of cancer cells to DNA damaging agents is most likely related to intrinsic deficiencies in DNA repair mechanisms. The capacity of cancer cells to recognize DNA damage and initiate DNA repair is a key mechanism for therapeutic resistance or recurrence. Here are a few questions for you to work out. 1. Comment on the damages caused to DNA due to oxidation of bases. 2. Elaborate the technique of adduct formation. 3. Discuss on damage caused by various ionizing radiations. 4. Discuss on human diseases caused by DNA repair system failure. 5. Describe human genes involved in DNA repair mechanism. 6. Regulation of DNA repair throughout the cell cycle. Explain. Here I can suggest a few books for your reference. Principles of Genetics, written by Gardner E.J., M.J. Simmons and P. Peter Snustad, 2009, published by Willy India Private Limited. Principles of Genetics, 7th edition, written by Robert H. Tamarin, 2002, published by Tata McGraw-Hill Education Private Limited. Molecular Biology, 3rd edition, written by David Friefelder, 1995, published by Narosa Publishing House, New Delhi. Principles of Biochemistry, 4th edition, written by David L. Nelson and Michael M. Cox, 2013. DNA damage and repair, mechanisms for maintaining DNA integrity, written by Clancy S., published in the journal Nature Education, 2008. Base excision DNA repair, written by Sarkov D.O., published in Cell Molecular Life Science in the year 2008. Here are a few websites for your further reference: www.personal.ksu.edu, www.ncbi.nlm.nih.gov/books, www.nature.com. Thank you for watching this program. Let's meet in the next session with a new topic. Until then, bye.